Assalamu alaikum wa rahmatullahi wa barakatuh, respected attendees. A'udhu billahi minash shaitanir rajeem, bismillahir rahmanir rahim. As Dr. Nusrat Saiba so kindly explained, today I'll be discussing boosting anti-tumor immunity with neoantigen vaccines, which are a relatively new form of cancer treatment. To start, however, I'd like to discuss the current state of cancer care and why it falls short. Traditional treatments have been around for many decades, and in fact prior to the 1970s, and many of us are familiar with these. They include surgery used to tackle resectable tumor masses, radiation, where high-energy waves like X-rays are used to shrink local tumor sites, and chemotherapy, where drugs can be delivered to have systemic effects on killing cells. More recent treatments have come out in the last decades and continue to be researched and studied, and these include hormone therapies, targeted therapies, immune checkpoint inhibitors, and others. Importantly, however, the trends in 5-year survival rates really vary across cancer types, highlighting the limitations of these treatments. For instance, we see that with prostate cancer, 97% of patients may be expected, statistically on a population-wide level, to have a 5-year survival rate. However, with something like pancreatic cancer, this number drastically falls, and it's just 13%. This is attributable to a number of reasons. For example, prostate cancer is often detected earlier, whereas pancreatic cancer is often detected at very late stages. And moreover, prostate cancer is more responsive to existing treatments, whereas pancreatic cancer is not. This is where neoantigen vaccines come in and where they may be able to help address limitations of existing treatments. Now, broadly, neoantigen vaccines fall under a category of therapies called immunotherapies, which, as the name suggests, are used to harness the body's natural defense system, its immune system, to kill tumors. With neoantigen vaccines, looking at this diagram on the right of the screen can be helpful, and I'll walk through it mainly in 3 steps to help explain what neoantigen vaccines are attempting to do. First, in our body, we have what are called antigen-presenting cells, T cells, and then antigens. Antigen-presenting cells, first, step 1, take an antigen, which is a small protein fragment, and portray it on their surface, shown here in pink. Then these antigen-presenting cells encounter inactive T cells, and these T cells are really our body's main attacking cell. Finally, when the antigen-presenting cell shows the antigen to a T cell and they bind, a T cell actually becomes activated, and then it's ready to go on and attack cells containing that specific antigen. The key innovation with neoantigen vaccines is this. If our antigen is a protein fragment from a tumor cell specifically, we call it a neoantigen. And if we use neoantigens to prime T cells, we can elicit T cell activity that's tumor specific, essentially using our T cells to specifically go and attack tumor cells as opposed to healthy ones. Now, neoantigen vaccine development really bridges computational research, wet lab research, and clinical care. And I think that's why it's really such a wonderful topic for biomedical engineering, because it does involve a spectrum of disciplines, including BME. On the in silico or computational side, researchers will first analyze tumor and normal DNA to identify neoantigens and candidate neoantigens. Secondly, computational algorithms are used to actually rank these neoantigens and determine which ones are most likely to activate T cells. Then in vitro, or essentially in a wet lab like a biology lab, researchers will test neoantigens in cell culture and actually validate whether or not they activate T cells. Synthesizing a vaccine with these neoantigens is also a critical step, and often there are additional components. These may include, for example, adjuvants, which are used to further stimulate the immune response. Finally, in vivo, neoantigen vaccines have to be tested in preclinical models, for example, in mice, and then, of course, progress on to clinical trials. So you may be wondering if these neoantigen vaccines work, and there really is a mixed body of research on this. The first clinical trial was held in 1997, and since then there's really been a flux of phase 1, 2, and even 3 clinical trials for these neoantigen vaccines. Across the board, what we see is that these vaccines tend to be safe and induce minimal side effects. And this is really promising because a huge lacking with current cancer therapies is that they induce many adverse side effects and promote nonspecific killing of both tumor and healthy cells. So with neoantigen vaccines, the fact that we see minimal side effects is a promising sign. On the other hand, if we're thinking about efficacy, how well neoantigen vaccines actually target tumors and shrink tumor masses, for example, results are mixed. Some patients respond very well, whereas others don't, and this can even be within the same tumor type and the same patient cohort. Therefore, there's really been a lot of interest in actually trying to characterize patient-specific immune responses and determine which immune characteristics may make patients more or less susceptible to these vaccines. Finally, I'd just like to end by really emphasizing that neoantigen vaccine development is an evolving and very active field of research, both on the in silico side as well as on the in vitro and in vivo side. In silico, there's a lot of interest in building better algorithms to rank neoantigens because current algorithms do fall short. They identify very few true positives, so in other words, very few neoantigens that really do elicit T-cell activity. In vitro, screening candidate neoantigens is a major task, and this is because it's very both labor-intensive and expensive to test hundreds of neoantigens. So figuring out efficient screening technologies is also important. And then finally, in vivo, there are many options for vaccine formulations in terms of what specific materials are used, what specific adjuvants, and so on. So characterizing and trying to understand which vaccine formulations work best is really important. And finally, just to reiterate, a lot of labs across the US are working on this. Alhamdulillah, I have the opportunity to work with Dr. Rachel Karchin at Johns Hopkins, and she's very interested, and we're very interested in the computational side of this problem. So we've been working on algorithms for neoantigen ranking. Finally, I'd like to end and bring this all back to a quote from the Holy Quran. So Allah Ta'ala tells us, Bismillah ar-Rahman ar-Rahim, "And there is not a thing, but with us are the treasures thereof, and we send it not down except in a known measure." And I chose to include this quote because I feel that neoantigen vaccines hold a lot of promise, but there are a lot of unknowns. And I think as researchers, we can humbly sort of think that Allah Ta'ala knows what will make the best vaccine or even the best cancer treatment. And we still don't know, so that's what we're trying to figure out and inshallah someday come to. So Jazakallah, thank you so much for listening.